Okay, welcome everybody. This is the second half of Chapter 22, and we're going to pick up with a discussion of adaptive immunity. Remember that when we talked about immunity last week, we noted that there's really two systems in place to keep you disease-free, and that's your nonspecific barriers, so your skin, your mucous membranes, and their secretions, the inflammatory response, and the granulocytes that basically patrol the tissues beneath ports of entry. And then, in the event that a pathogen makes it past those barriers, we have our adaptive immunity, which will generate a chemical and physical attack on the pathogen based on its specific characteristics. So it involves the ability to recognize, respond to, and remember a particular substance, and this is stimulated by the presence of antigens, which are large molecules that are not similar enough to those that are produced in the body um, for the immune system to tolerate them. They're introduced from the outside, and they include um, proteins, polysaccharides, lipids, or combinations of these molecules that are unique to the strain of pathogen that brings them in. Examples of components that can contain antigens include pollen, animal dander, feces, mites, food, and drugs. All of these can evoke an immune response. Self-antigens are made by the body, and they're used as markers to allow adaptive immune response to differentiate the cells of your body from those that come in from the outside. And so one of the things we have to appreciate about adaptive immunity is that it's not there to evaluate beneficial versus harmful. It's there to distinguish self from non-self. The response to self-tumor antigens is an example, and the response to self-antigens resulting in tissue destruction, such as seen in autoimmune diseases, is another example of the immune system's interaction with these antigens. Haptens are tiny molecules that combine with large proteins and produce an adaptive immune response. Um, there are two basic types of immune response. There's the humoral immunity, which is mediated by B cells, and there's cell-mediated immunity, which is the purview of the T cells. And we'll find out when we get into a little bit more detail that the T cells can be broken down into regulatory, cytotoxic, and helper T cells, all with distinct roles in this adaptive immunity. Now, this is just a chart, 22.3 in your book, showing you a comparison of innate and adaptive immunity. I won't go through each component here, but again, star this slide. This is excellent quiz and test material. So let's talk a little bit about where the lymphocytes come from. Positive selection ensures survival of lymphocytes that react against antigens and then proliferate and form clones. And this is a necessary characteristic for lymphocytes because they have to have the ability to, upon interaction with a presented antigen, become stimulated to multiply and generate an army capable of destroying anything that's attached to that particular antigen. Negative selection eliminates clones of lymphocytes that react against self-antigens. And the idea here is that we don't want to retain any lymphocytes that would recognize, bind to, and initiate an attack on our body's own tissues because that would produce damage. Tolerance is essentially the state of unresponsiveness of lymphocytes to a specific antigen, usually to the antigens that are on our own cells. Many different kinds of clones exist because of the genetic recombination that occurs during development. We have close to 1 billion different specificities for the B and T cell populations. The B and T cells originate in the red marrow and move to primary lymphatic organs where they mature into functional cells. The B cells move to the bone marrow and the T cells move to the thymus. And as you can see, those designations, B and T, are essentially the locations where they become immunocompetent. Secondary lymphatic organs and tissues are where the lymphocytes are going to interact with each other and antigen presenting cells and antigens in order to generate the immune response. We have diffuse lymphatic tissue, lymphatic nodules, tonsils, lymph nodes, and spleen that all represent secondary lymphatic organs. So basically what happens is that we generate these B and T cells in the red marrow. We chemically educate them in the primary lymphatic organs, and then once they're capable of recognizing, binding to, and attacking any particular pathogen that's bound to an antigen, they move out to their secondary locations. Lymphocytes have to be able to recognize a foreign antigen, and after recognition, the lymphocytes have to increase in number in order to effectively destroy it. So what you're looking at here is just sort of a schematic showing you what an antigen might look like. These different shapes are the basis for the interaction between the lymphocytes and the presented antigens. Antigenic determinants are specific regions of an antigen that are recognized by a lymphocyte. Activation occurs by antigenic receptors. These would be T and B cell receptors on the surface of lymphocytes that combine directly with the antigenic determinant, or by activation involving glycoproteins, which are called major histocompatibility complex molecules that are attached to the plasma membrane and have a variable region that can bind to foreign and to self antigens. So let's take a look at antigenic determinants.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
Okay, the major histocompatibility complex comes in two classes. Class 1 are found on the surface of all nucleated cells and in concert with antigens that were produced inside the cell from, for example, digested virus particles. This is like displaying a flag that says, "Kill me." MHC restricted are both MHC1 and foreign antigen displayed together. Class 2 are found on the surface of an antigen-presenting cell. The B cells, the macrophages, the monocytes, and dendritic cells are all examples of cells in this class. They display MHC2 with foreign antigen, and this is a signal that says rally round the flag. It stimulates other immune system cells to respond to that particular antigen. So let's take a look at how antigen processing operates.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class 1 MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class 1 MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the Class II MHCs, and the two structures combine. The MHC Class II antigen complex is transported to the plasma membrane. The displayed MHC Class II antigen complex can stimulate other immune system cells to respond to the antigen.
Okay, co-stimulation is essentially a method for promoting the proliferation of a selected B or T cell based on its ability to interact with that particular antigen. In order for B or T cells to produce a response, there has to be binding of the MHC class II antigen complex to a T cell receptor, followed by co-stimulation, which occurs via cytokines. These are essentially soluble molecules released by a macrophage that binds to a receptor on the helper T, and also by surface molecules that bind molecules. Examples include the B7 and the C28 on the macrophage and helper T, and this holds the cell together. So what you're doing in this process is you're bringing the antigen presenting cell and the cell to be stimulated in close proximity. You're making certain that you're stimulating the proper cell, both as a result of the receptor and presented antigen interaction, and also by these other cell surface molecules that serve as a second check to make certain that the cell we're stimulating is in fact a lymphocyte, and in addition to that does have the specificity that we're calling for by presenting that particular antigen on the surface. If co-stimulation doesn't take place, the cell will go into anergy, where essentially it'll be alive but unresponsive. This is the usual situation where anergy occurs when T cells encounter self-antigens. So this is just a look at how co-stimulation operates. The helper T cells are activated by a first signal and by co-stimulation. The first signal is the binding of the MHC class II antigen complex to the T cell receptor. Co-stimulation is an additional signal, such as molecules released from another cell. For example, macrophages release cytokines that bind to receptors on helper Ts, resulting in the co-stimulation. You can see here the macrophage with the presented antigen, and the helper T recognizing the presented antigen, but also binding to the cytokines that are produced by the macrophage, and this is going to stimulate now proliferation of, and I can't stress this highly enough, a particular subpopulation of helper T cells, as opposed to every helper T cell in the body. This is what makes adaptive immunity so effective. Other co-stimulatory signals are the combining of surface molecules between cells, such as the binding of a B7 molecule of the macrophage with a CD28 molecule on a helper T. The CD4 molecule on the helper T binds to the macrophage's MHC class II molecule, and it helps hold the cell together. Okay, so you can see here how, again, the presented antigen provides the specificity for interaction, and the CD4, which is part of the cell surface proteins on the helper T, also is capable of interacting with the MHC, and we got a secondary check as a result of the interaction between the CD28 of the helper T and the B7 of the macrophage. So this is, again, a type of handshake that ensures two things. Number one, that we are in fact having an interaction between, in this case, a macrophage and a lymphocyte, and number two, that the interaction is with the particular lymphocyte that's going to recognize, bind to, and destroy anything attached to that particular antigen. Okay, cells from the original clones have to proliferate before antigen can be effectively attacked. You have to get your numbers up in order to be capable of wiping out the pathogen, and this requires that we have proliferation of helper Ts and B and effector Ts as well. So what you're looking at here is just a flow chart showing you how we stimulate the proliferation of B cells. Prior to B cell activation by a helper T, the B cell has to endocytose and process the same antigen that activated the helper T. This antigen binds to a B cell receptor, and both the receptor and the antigen are taken into the cell via endocytosis. The B cell then uses an MHC class II to present the processed antigen to a helper T, whose receptor binds to the MHC class II antigen complex. Co-stimulation of the B cell by a CD4 surface cell protein and other surface molecules also occur. So you can see here again the helper T CD4 interacting with the MHC class II. Again, a way of sort of securing this chemical handshake. Co-stimulation by interleukins. These again are diffusible compounds that promote cell division, are released from the helper T cell, causing it to divide, and the resulting daughter cells divide and soon produce many cells that are capable of generating large amounts of antibody. The antibodies are part of the immune response that's going to attach to, bind, and destroy the antigen. Okay, and the reason that we call this entire process humoral immunity is because it's mediated in the body fluids. The B cell can be quite some distance from the pathogen or antigen that it's designed to attack, and the antibody essentially upon connecting with the foreign antigen that it's designed to recognize will cause destruction of anything attached to that antigen by a variety of processes. There will be complement fixation and lysis in some cases. There can be agglutination. There can be immunoprecipitation. There can also be simply masking of compounds such as endotoxins and exotoxins. So this table is just showing you the different immune system cells and their primary functions. Again, I won't go through each of these. Just star this slide, know this material. Again, this is excellent test and quiz material. So let's take a look at T cell dependent antigens.
Proteins generally require the cooperation of T helper cells, Th2, to stimulate B cells into becoming antibody-producing cells and memory cells. Such antigens are therefore said to be T cell dependent. The protein antigen is first ingested and then broken into peptide fragments by an antigen-presenting cell, such as a macrophage. The antigen is then moved to the surface of the macrophage on a class II MHC. The T cell receptor on the surface of a T helper cell recognizes the peptide antigen that is being presented on a class II MHC of the macrophage, and the T helper cell becomes activated. The activated Th2 cell is now capable of stimulating B cells. B cells also ingest antigens. The antigen reacts with an antibody on the surface of the B cell and is then internalized, digested, and presented at the surface of the B cell on a class II MHC. The antigen class II MHC complex of the B cell interacts with the T cell receptor on the activated T helper Th2 cell. The T helper cell produces cytokines, which stimulate the B cell to proliferate and differentiate into antibody-producing plasma cells and memory cells. Antigens, such as bacteria or viruses, are usually ingested and degraded into small fragments by phagocytes such as macrophages. The small fragments are then complexed with a special type of protein called a class II major histocompatibility complex protein, MHC, and transferred to the surface of the phagocytic cell. Helper T cells recognize an antigen when it is presented on a class II MHC because helper T cells have special recognition proteins on their surfaces called T cell receptors. This interaction activates the helper T cells. The activated helper T cell is now capable of stimulating B cells to become antibody-producing plasma cells.
Okay, we also have to keep a brake on lymphocyte activity. We don't want lymphocytes to recognize anything outside foreign antigens. If so, there's going to be the potential to attack your body's own tissues, and if that occurs, we'll have a condition that we classify as autoimmunity. So tolerance is simply the state of unresponsiveness of lymphocytes to a specific antigen. Most common is the unresponsiveness to self-antigens, and this is provoked by deletion of self-reactive lymphocytes during the formation of the immune system and the prevention of activation of lymphocytes that encounter self-antigens. Also, activation of suppressor cells can produce suppressive cytokines or kill antigen-presenting cells, but their method is poorly understood. However, we think that defects in these processes lead to the phenomenon in some cases that we classify as autoimmunity, where your immune system turns about and attacks your body's own tissues in a misguided attempt to defend you against disease. Antibody-mediated immunity is effective against extracellular antigens, including bacteria, viruses, protozoans, fungi, parasites, and toxins when they're outside the cells. Antibodies come in various classes based on the composition of their constant regions, and the classes are IgG, IgM, IgA, IgE, and IgD. The structure includes a variable region, which is the part that recognizes and binds to a particular antigen. That's the part that comes in a billion different flavors when we're referring to the T and B cells. And then the constant region, which is responsible for activities of antibodies such as activating complement or attaching to different types of white cells. So these constant regions provide either a way to staple the antibody to a tissue or to a membrane, or to allow it to float freely in tissue fluids, or to dimerize or pentamerize or trimerize, depending on, again, the antibody class. And so what you're looking at here are some of the different antibody classes. Now, they're all the same in the sense that they recognize and bind to foreign antigens. Where they differ is where you find these antibodies primarily, okay, and essentially uh, where they're most likely to encounter pathogen. So if we look at IgG, these activate complement and promote phagocytosis. They can cross the placenta and provide immune protection to the fetus and the newborn, and they're responsible also for a phenomenon called Rh incompatibility. Examples of this include hemolytic disease of the newborn, and in some cases we also see this in mismatched transfusion reactions. So you can see what the IgG looks like. Okay, it's a relatively simple Y-shaped molecule, two heavy chains, two light chains. The antigen binding regions are at the tips of the Y, and the constant regions are basically the areas that are going to essentially be the same regardless of the specificity of the IgG antibody. IgMs activate complement and act as antigen binding receptors on the surface of B cells, and they're responsible for transfusion reactions in the ABO system. They're often the first antibody produced in response to an antigen. And then the IgAs are secreted in saliva, in tears, and mucous membranes. They provide protection on body surfaces, and they're found in the colostrum of milk. It provides immune protection to newborns. So this is an example of an interesting type of immunity that we call natural passive immunity. And we'll go into that in just a bit. IgE binds to mast cells and basophils and stimulates the inflammatory response. You can see IgE here, again, this simple Y shape. And IgD functions as an antigen binding receptor on the B cell. And again, the reason for these different classes is the difference in the constant region. Remember that there's a constant region in both the light and the heavy chain, and within a class, the constant region will not vary. The variable part, again, is the antigen binding region at the tips of the Y-shaped molecule. So how do antibodies essentially attack and destroy antigens, or to be more precise, pathogens that are attached to the antigens? Well, they can inactivate the antigen, where the antibody binds to the antigen and inactivate it basically by cloaking the antigen and rendering it incapable of producing disease. They can bind antigens together. This is a phenomenon called immunoprecipitation. Essentially, what happens is you produce a complex that has such a large molecular weight that it comes out of solution and becomes an easy target for a macrophage, which can come by and destroy it. And then it can also activate the complement cascade that leads to essentially membrane rupture and lysis. So one of the things that occurs when the antibody attaches to the antigen that's attached to the pathogen is that the complement proteins will interact with the constant regions of the antibody, and this stimulates inflammation, chemotaxis, and also lysis. Okay, again, to review, inflammation we've already talked about previously, but just to review, that's the phenomenon where the area of the injury or the infection becomes reddened, it swells up, becomes edematous, it warms up, and it becomes painful. And the idea behind inflammation, again, is to bring blood to the area, and with the blood come the white blood cells, which are the disease fighters. Chemotaxis is simply the movement of, in this case, white blood cells towards diffusible chemicals that are produced in the area of the injury or the infection that draw the white blood cells to the area where they can do the most good fighting the pathogen. And of course, lysis is simply cell rupture. What else can antibodies do to bring about the destruction of pathogen? Well, they can initiate the release of inflammatory chemicals. An antibody binds to a mast cell or a basophil, and when an antigen binds to the antibody, it triggers the release of chemicals that will cause inflammation. And they can also facilitate phagocytosis. Basically, by tagging the pathogen, we draw phagocytic cells to the region. They then endocytose the pathogen, chop it into little bits, and that performs two tasks. The first task is that it destroys the pathogen. The second thing that's accomplished is that components of the digestive pathogen are presented on the membrane surface as a warning to the immune system that we have an infection, and we can now stimulate our specific immunity even further. If we look at antibody production, what we see is two distinct profiles um, that are going to differ depending on whether it's our first or second exposure to a particular antigen. The primary response happens when a B cell is first activated by an antigen. The B cell proliferates to produce plasma cells and memory cells. Now, these memory cells are very interesting because what they do is they reside in the body for decades, and in the event that the pathogen, the exact same strain of pathogen, shows up a second time, the numbers of B cells are so high that we can initiate an immune response that essentially destroys the pathogen in short order, and as a result, we rarely succumb to disease. These memory cells are stored every time you essentially fight off a disease state and as a result of an immune response. And this is one of the reasons why often if you were extremely sick as a child, you tend to be relatively healthy as an adult simply because of the large number of memory cells that you have stored in your lymphatic organs. The secondary response occurs during later exposure to the same antigen. These memory cells are basically a standing army ready to generate plasma cells and additional memory cells the second time that exact same strain of pathogen shows up. The result is that we generate much more antibody much more quickly. We neutralize the pathogen, and the result is essentially no chance to generate disease. Cell-mediated immunity is the purview of T cells. They are most effective against intracellular microbes, viruses, fungi, intercellular bacteria, and also parasites. Cytotoxic Ts function in two ways. They lyse virus-infected cells, tumor cells, and tissue transplants. Major lysine is perforin, which forms a hole in the plasma membrane of the target cell. We call this a membrane attack complex. They produce cytokines, which promote phagocytosis and inflammation. Again, that's going to allow us to consume and destroy the pathogen, and the inflammation, of course, brings blood to the area. As in antibody-mediated immunity, memory cells are also produced. Delayed hypersensitivity in T cells is an example of cell-mediated immunity that is somewhat misdirected. It's involved in allergic reactions such as exposure to poison ivy or poison oak. In this phenomena, the hapten binds to proteins in the skin, and then the antigen processed by the Langerhans cells of the skin present it, and this results in delayed hypersensitivity in the T cells, because what happens is that we produce now histamine release as a result of mast cells releasing the inflammatory chemicals, and we generate now the characteristic blisters, reddening, and itching that we associate with exposure to these types of haptens. This is just a look at how cytotoxic T cells are stimulated to proliferate. It's very similar to the way that B cells are stimulated to proliferate. An MHC class 1 molecule displays an antigen like a viral protein on the surface of the target cell. The activation of a cytotoxic T begins when the T cell receptor binds to the MHC class 1 antigen complex. There's co-stimulation of the cytotoxic T by CD8 and other surface molecules, and co-stimulation by cytokines such as interleukin-2, which are released from helper Ts. The activated cytotoxic Ts then divide, the resulting daughters divide, and so on, and eventually we generate an army of cytotoxic Ts capable of going out and destroying essentially infected cells or pathogens. And so let's take a look at how cytotoxic T cell activity acts on target cells.
When a virus or other infectious agent infects a cell, some of the proteins of the infectious agent are broken down into peptide fragments. These fragments are complexed with special proteins called Class I MHCs, major histocompatibility complex proteins, and displayed on the surface of the infected cell. The T cell receptor on the cytotoxic T cells interacts with the virus-infected cells by recognizing both the foreign protein fragment and the Class I MHC. The cytotoxic T cell then releases toxic substances which cause death of the infected cell. Cytotoxic T cells also produce a toxin called perforin, which creates holes in the cell membrane, causing the cell to break open.
Okay, acquired immunity is essentially the result of exposure to antigen. We can acquire immunity as a result of our activities of daily living, or we can have deliberate exposure to an antigen via phenomena such as vaccination. There's different classes of acquired immunity. We can have passive immunity, where essentially we rent an antibody from another source, and this provides temporary resistance to the disease for the amount of time that the antibody is still in our system. Eventually, it'll be processed out and we'll become susceptible again. And then we have active immunity, where we actually produce the B and T cells capable of fighting the pathogen in our own immune system. And this is actually a more long-term and more efficient way of protecting us against disease, because this is the type of immunity that will stay with you your entire life. In immunization, we have deliberate exposure to the antigen or antibody. Active natural immunity is the result of natural exposure to an antigen. Active artificial immunity is the result of vaccination, where we deliberately expose ourselves to an antigen through essentially an injection. Okay, so Active natural, you pick it up as a result of your activities of daily living. Active artificial, you get a vaccination. Passive natural is the transfer of antibodies from a mother to her fetus or to her baby. And passive artificial immunity is an injection of antibody in order to provide temporary immunity to a particular disease. Antisera are available for things like rabies, hepatitis, measles, and bacteria such as tetanus and diphtheria, and also venoms from snakebite and black widow spiders. What you're looking at here are different methods to acquire adaptive immunity. And so we'll start down here, the left-hand side of the flow chart. Natural immunity is where the antigens are introduced as a result of natural exposure. Okay, artificial is where the antigens are deliberately introduced via an injection. Okay, the result here is upon exposure to the antigen, the immune system will produce an army of B and T cells that are capable of recognizing and destroying anything that that antigen is attached to. The result here is acquired adaptive immunity that will last throughout your lifetime. Okay? In passive immunity, what happens is that we rent antibodies from another source. In natural passive immunity, what happens is, for instance, a child in utero can receive antibodies from the mother's immune system to provide temporary protection against pathogens until the child's immune system is immunocompetent. Or in the nursing infant, we can transfer antibody via the colostrum in the mother's milk that provide prophylactic protection until the child's immune system is capable of fighting disease on its own. Okay, so that would be natural passive immunity. It's a temporary situation. Artificial passive immunity is where you receive antibody as a result of injection that'll provide temporary resistance to disease for the amount of time that the antibody stays in your system. Eventually what will happen in both natural and artificial immunity is that those antibodies will disappear, they'll be processed out, and you will become susceptible again. The passive immunity will be transferred from another person or an animal to the individual receiving it. This results in acquired adaptive immunity. In the passive immunity, it's short-term. In the active immunity, remember that this is a long-term situation.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Okay, let's take a look at the immune response. Okay, now as we age, of course, the immune system does not improve, right? Primary and secondary responses decrease. More antigen is required to produce a response, so we have a slower response to pathogen. We produce less antibody. We generate fewer memory cells. The ability of cell-mediated immunity to resist intracellular pathogens decreases. And we have reactivation of pathogens, such as herpes zoster, which is the agent of chickenpox, which lives in the dorsal root ganglia, and when the immune system is incapable of keeping the zoster in check as it erupts from the nerve endings, what can happen is in the adult we have a condition known as shingles. This is one of the reasons why we promote vaccination against herpes zoster in individuals that are, say, 65 and older, as a way to sort of remind the immune system that you have this DNA latent inside your nerve cells that at any time can essentially start producing zoster pell-mell, and the eruption through the nerve endings is what produces those blisters and those pustules that produce the manifestations of the disease. Other things that occur as the immune system ages is that we see higher incidence of autoimmunity, and obviously we have decreased ability to resist infection and disease. Okay, that brings me to the end of this block on 22. Again, review this material as it will be covered on PRS quiz and the exam, and I'll see everybody in class.